I am going to talk about polydrymnus, transient antenatal Bartter syndrome, and MAGE2 mutations. But first of all, I am going to explain what is the Bartter syndrome. It is a rare genetic defect in the thick ascending limb of the loop of Henle. It is characterized by hypokalemia, metabolic alkalosis, and normal or low blood pressure. There are two different types of Bartter syndrome, the neonatal and the classic. The Gentleman syndrome it is a disease that is closely related to this syndrome, with intermediate characteristics between the two types.
Hello friends, today we'll be discussing two important causes of chloride unresponsive hypokalemic metabolic alkalosis. The two causes are Bartter syndrome and Gentleman syndrome. Now these two syndromes share a variety of common features such as a low potassium, metabolic alkalosis. The BP is usually normal to slightly low. Both these patients have tertiary hyperaldosteronism, which is due to the sodium loss in urine, and this manifests as an increase in plasma renin and aldosterone levels. Both these patients also have low levels of magnesium in their serum. Magnesium levels, however, are markedly low in gentlemen, which is an important differentiating point between Bartter's and Gentleman's. Now, about the pathogenesis, Bartter syndrome occurs due to the mutation of sodium potassium chloride cotransporter present in the thick ascending limb of loop of Henle. Due to the impairment, there is increased loss of sodium, potassium and chloride in urine, as well as an impaired reabsorption of calcium. This causes increased loss of calcium in the urine, leading to hypercalciuria. In gentlemen, there is an impairment of sodium chloride cotransporter present in the distal convoluted tubule, and a contrasting point with Bartter's, this sodium chloride cotransporter impairment actually leads to increased reabsorption of calcium, thereby presenting as hypocalciuria.
In the background, there were 3 pregnancies with male offspring in one family were complicated by severe polydrymnus and prematurity. One fetus died, the other one had transient massive salt wasting and polyuria reminiscent of antenatal Bartter syndrome.
Babies in the uterus are surrounded by amniotic fluid. However, some women do develop increased amounts of this amniotic fluid as the pregnancy progresses. And at around 30 weeks, some women are found to have excessive amounts of amniotic fluid around the baby, and this is called polyhydramnios. Now, it might be felt by the midwife on palpation, or it might be diagnosed on scan. But certainly, depending on what the findings are, would determine the management from then on. In the majority of cases, polyhydramnios will resolve itself and the woman goes on to have a perfectly normal, healthy baby. But polyhydramnios in some cases can show an indication that there is a problem with the baby. So it might be that the woman is sent for further tests. She might need to have an amniocentesis. The mode of delivery again might come into question depending on what the findings are. With polyhydramnios, because of the amount of fluid that is circulating round, there can be a risk of the baby's cord coming down in front of the baby's head if the waters break. So again, it would be very much on an individual basis how it would be managed.
To uncover the molecular cause of this possibly X-linked disease, they performed whole exome sequencing of DNA from 2 members of the index family and targeted gene analysis of other members of this family and of 6 additional families with affected male fetuses. Women with idiopathic polydramnios who were pregnant with male fetuses were evaluated too. They performed immunohistochemical analysis, knockdown and overexpression experiments, and protein-protein interaction studies.
Do you perform qPCR as a routine part of your research? Are you interested in gene expression but haven't yet looked into how next-generation sequencing or NGS can help you out? Let me see if I can help answer some of these questions. Next-generation sequencing, also known as NGS, provides opportunities to profile gene expression in a wide range of settings. Whether you're looking to discover potentially novel genes or transcripts, or study how certain genes respond to stimuli, NGS can offer you a great complement to your existing methodology. Let's say you're interested in studying a particular set of genes' response to a certain stimulus, and certainly you're using a set of TACMAN probes to accomplish this. This works well enough, but you're relying on prior knowledge of this particular set of genes' involvement in the affected pathway. Are there other genes or RNA transcripts that are playing a part in the response? Let's think a bit differently and talk about targeted RNA sequencing. Using this approach, you can target a known set of genes or non-coding RNAs and measure their expression levels, much like a qPCR or TACMAN approach. This method can allow you to target a small number of custom-selected genes with custom ion AmpliSeq RNA panels, or all the way up to over 20,000 genes in a single reaction using ion AmpliSeq transcriptome, for instance. These methods also allow for low input starting material and work well with degraded RNA, such as that sourced from formalin-fixed paraffin-embedded tissue, also referred to as FFPE. When combined with the ability to multiplex many samples and sequence them together in a single run, this approach provides the ability to economically analyze a large number of targets within a large number of samples. For targeted RNA sequencing, analysis is much more straightforward. Data is analyzed using a provided software plugin available in Torrent Suite software, and gene expression values are provided as well as clustering of genes that exhibit different patterns of expression. The expression data can be used in a variety of standard software packages for further statistical analysis if desired. For many, this is the best of both worlds. Being able to use low amounts of input material, having a comprehensive set of genes or transcripts to target economically, and have a simple analysis path to allow them to move right into the interpretation and understanding of their data. I hope this video was helpful regarding gene expression applications using next-gen sequencing, but I'm sure you'll have more questions. So submit your questions at thermofisher.com/ask and subscribe to our channel to see more videos like this. And remember, when in doubt, just seek it out.
In the results, they identify a mutation in MAGE2 in the 30 infants in analysis who had transient antenatal Bartter syndrome, 2 mutations in 2 families with idiopathic polydramnios, and 4 patients died perinatally and 11 survived. All symptoms disappeared during follow-up in the infants who survived. They showed that MAGE-D2 affects the expression and function of the sodium chloride cotransporters NKCC2 and NCC, key components of salt reabsorption in the distal renal tubule, possibly through adenylate, cyclase, and cyclic AMP, signaling and a cytoplasmic heat shock protein. In conclusion, they found that MAGE-D2 mutations caused by X-linked polydramnios with prematurity and a transient form of antenatal Bartter syndrome. MAGE-D2 is essential for fetal renal salt reabsorption, amniotic fluid, and homeostasis, and the maintenance of pregnancy.
Bartter syndrome is a rare inherited defect in the thick ascending limb of the loop of Hing. It is characterized by low potassium levels, increased blood pH, and normal to low blood pressure. There are 2 types of Bartter syndrome, neonatal and classic. A closely associated disorder, Geit-Hellman syndrome, is milder than both subtypes of Bartter syndrome. Features: in 90% of cases, neonatal Bartter syndrome is seen between 24 and 30 weeks of gestation with excess amniotic fluid. After birth, the infant is seen to urinate and drink excessively. Life-threatening dehydration may result if the infant does not receive adequate fluids. About 85% of infants dispose of excess amounts of calcium in the urine and kidneys, which may lead to kidney stones. In rare occasions, the infant may progress to renal failure. Patients with classic Bartter syndrome may have symptoms in the first 2 years of life, but they are usually diagnosed at school age or later. Like infants with the neonatal subtype, patients with classic Bartter syndrome also have polyuria, polydipsia, and a tendency to dehydration, but normal or just slightly increased urinary calcium excretion without the tendency to develop kidney stones. These patients also have vomiting and growth retardation. Kidney function is also normal if the disease is treated, but occasionally patients proceed to end-stage renal failure.